Hello everyone and welcome back to the new session. Myself Dr. Narkade Gopal Vasudev, working as Assistant Professor in Department of Genetics and Plant Breeding, School of Agriculture, SR University, Warangal. Today's session topic is advances in citrus breeding. So, botanical name, citrus species, family, Rutaceae, chromosome number twice n is equal to 18. So citrus constitutes a major group of fruits comprising mandarins, oranges, lemons, pomelo, grapefruit, tangelo, trifoliate orange, citron and citronges. Despite of interspecific and intergeneric hybrids, Poncirus and Fortunella also belongs to genus Citrus. During its long history, citrus has given the world numerous varieties, both by open pollination, bud sports, and of recently by controlled pollination and artificial induction of bud variation. India is considered to be the home of several citrus species, and they are found growing wild in some parts of the country. Many types of citrus still remain unexploited by the man, and such types are considered as semi-wild. Then, center of diversity. So, there are three major centers of diversity in India. The first in the northeast, including Assam and adjoining areas. The second, diversity in South India. Indigenous types include Gajanimma, Kichili and some wild mandarin types. The third, northwest region at the foot of Himalayas where the hill lemon, that is galgal, is common. Two species of subgenus Eucitrus, that is Citrus indica and Citrus assamensis, and three species of subgenus Papeda, that is Citrus latips and Citrus ichagenensis and Citrus macroptera are native of Assam region. Citrus medica is said to be the native of northwest of India. Lemon is sometimes cited to be indigenous to India, but its actual home is Malaya region. The sweet oranges are considered to be the native of southern China and then spread to India. Washington Navel, Brazil, limes, East India, grapefruit, West Indies, mandarin, Indochina, kumquats, South China, trifoliate orange, central and northern China, microcitrus, Australia, Eremocitrus, Australia, and that Eremocitrus is xerophytic. Then next, classification of citrus species. So majorly there are different types of groups like acid group, mandarin group, pomelo and grapefruit group, orange group, papeda group and other species group. So in acid group, acid lime, Citrus aurantifolia, sweet lime, Citrus limetoides, Persian lime, Citrus latifolia, Rangpur lime, Citrus limonia, lemon, Citrus lemon, rough lemon, Citrus jambiri, sweet lemon, Citrus limetta, Volcamer lemon, that is Citrus volcameriana, Kharna khatta, Citrus kharna, citron, Citrus medica. So all these species belongs to acid group. Next, mandarin group, that is Santra or sweet orange, that is Citrus reticulata. Satsuma mandarin, Citrus unshu. Willow leaf mandarin, Citrus deliciosa. Cleopatra mandarin, Citrus reshni. King, Citrus nobilis. Tangerines, Citrus tangerina. Temple, Citrus temple. So this species belongs to mandarin. Now, pomelo and grapefruit. That is, pomelo means Citrus grandis or Citrus maxima. Grapefruit, Citrus paradise. And kumquat, Fortunella species. Next group, orange group. That is sweet orange, Citrus sinensis. Sour orange, Citrus aurantium. Myrtle leaf orange, Citrus myrtifolia. Then Japanese summer grapefruit, Citrus natsudaidai. Next group, papeda group. Chang papeda, Citrus ichangensis. Uzu of Japan, that is Citrus jumos. Khasi papeda, Citrus latips. Melanesian papeda, that is Citrus Macroptera, Mauritius papeda, that is Citrus hispix, and Alamo, that is Citrus macrophylla. Other species group, that is Kichi, Citrus maderabstana, Calamondrin, that is Citrus madurensis, Indian wild orange, Citrus indica, Gajanimma, that is Citrus pennivesiculata. So, these are the different classification of citrus species. Next, Tanaka classification genus citrus into two subgenera, namely Archicitrus and Metacitrus. So, the genus citrus includes two subgenera and 144 species. In Archicitrus, 5 sections, 98 species. That is, Papeda include 12, Limonellus includes 16, Arantinum includes 28, Citrophorum include 21, and Cephalocitrus includes 21 species. Metacitrus include 3 sections and 46 species. Osmocitrus 9 species, Acromen a big section and has a 36 species. Pseudofortunel has only 1 species. So now we will see the history of improvement. Like in USA, systematic experiments planned by W.T. Swingle and H.J. Weber in the year 1892 to 1893. In Japan, work started in 1909. So in India, work hybridization started in 1912. In Philippines, hybridization started in 1928. In USSR, started extensive crosses in 1930. In Spain, 1932, and in Java, 1928. So next, most important thing, that is germplasm resources. The exotic collection of citrus germplasm was started in the year 1940. Kinno mandarin was one of the collections which is now a leading cultivar in northwestern India. Besides, other exotic collections were Valencia lead, Washington navel, Jaffa, Malta blood red, pineapple, ruby orange, Satsuma, Dancy tangerine, Clementine and Cleopatra, Wilking, Temple, Duncan, Mars seedless, Lesbon lemon, trifoliate orange, Dancy trifoliate orange. More than 650 accessions are being maintained at CHES Chethali Bangalore, CHES Ranchi, NRC on Citrus Nagpur, Horticulture Experiment Station at Bhatinda, IRI New Delhi, MPKV Rahuri, Citrus Improvement Project Tirupati, Citrus Experimentation Station at Nagpur, Citrus Experiment Station in Assam. So here different accessions are being maintained. So during 1988, as a result of systematic exploration by NBPGR in northeastern region, Citrus indica and many endangered species were collected for conservation. Northeastern region is a hunting ground of biodiversity of citrus species. Chakravar et al. In 1988 identified two promising clones of acid lime, that is Vikram and Pramalini in Maharashtra. At Nagpur, seedless Santra has been selected, which gives high yield and quality fruits. Attempt has been made during 1978 by NBPGR to preserve Citrus indica, which is the progenitor of Citrus reticulata. For establishment of gene sanctuary, National Park, the national genetic diversity of Citrus indica was observed in the forest of Garo Hills in Meghalaya. The natural genetic diversity of Citrus indica was observed in forest of Garo Hills in Meghalaya, which exhibited the plant characters varying from bush to climber with high frequency of distribution in dense forest and showing resistance to biotic stresses. Therefore, a gene sanctuary for Citrus indica was established in Tanga Range in Garo Hills. The genetic material of citrus is conserved in field gene bank or repository. The next most important aspect, that is problems in citrus breeding. So as we are dealing with the advances in citrus breeding, so now we will see what are the different problems. So first is time. Citrus being perennial in nature, it takes more time for bearing. However, this period can be reduced to a maximum of half by top working the seedlings on an old tree. Next problem, that is polyembryony. It is a peculiar feature found in citrus in which seed consists of more than one embryo. For getting large number of hybrids, citrus breeders should select a seed parent known to be either monoembryonic citrus species or polyembryonic, except Citrus medica, Citrus latifolia and Citrus grandis, which are monoembryonic, restrict to the choice of breeder and complicate the procedure required to attain the desirable objective. Then third, sterility. Prevalence of high generative sterility is obviously a serious hindrance in the use of a particular parent for hybridization. Complete pollen sterility is problematic, where proportion of nucellar embryos are very high. High level of sterility often leads to production of seedless fruits, which is serious hindrance to develop the varieties. Then fourth one, self-incompatibility. Self-incompatibility and cross-incompatibility is a common phenomenon which occurs widely in citrus. Most of the varieties of grapefruit, that is Citrus grandis, are found to be self-incompatible. Besides, some varieties of lemon, sweet orange, mandarins exhibit self-incompatibility of gametophytic type, governed by oppositional alleles. Sweet orange varieties like Washington Navel and Satsuma mandarin are having sterile pollen. Thereby, they produce parthenocarpic fruits if cross-pollination is not done through viable pollen. Next problem, that is long juvenility. It is a major barrier in the progress of citrus breeding in India. General treatments to shorten the period or induce early flowering have not been generally effective. It was reported that neither chemical treatments nor incorporation by genetic transfers has been effective in combating the long juvenility in citrus. Now, what are the breeding objectives? Breeding objectives of citrus. First, producing early maturing citrus fruits with high yield and fruit quality. To breed varieties suitable for different agroecological regions. To develop varieties having less tendency of granulation. To breed varieties suitable for export. In rootstock breeding, the main emphasis has been given on the development of rootstock resistant to Tristeza virus, that is Phytophthora and nematode. Most of the breeding programs make use of Poncirus, which is a carrier of resistance to Tristeza, Phytophthora and nematode, besides cold hardiness. So mainly we need to concentrate on Tristeza virus, then Phytophthora. So these are the major things we need to concentrate. Then salt tolerant rootstocks have been found Salt tolerant rootstocks have also been found possible in some progenies involving Cleopatra and Sunki mandarin and Rangpur lime. Next we will see floral biology. So flowering in citrus takes place during Feb to April. In North India, sweet orange and mandarins bloom only once, that is in March. However, it is reported that sweet oranges bloom twice in a year under Bihar conditions, once in February-March and another that is in June-July. Inflorescence in citrus species is of cymose type. Generally, anthesis takes place in the morning between 9 to 12. Flowers on the shaded side of the tree have been observed to open later than exposed to sunshine. Dehiscence of anther takes place at different times, that is in lemon before anthesis and in before sweet orange, sweet lime after anthesis. Oozing of gummy substance on the stigmatic surface is the indication of stigma receptivity. In various citrus varieties, receptivity starts 2 to 3 days earlier to anthesis and lasts 4 days after anthesis with maximum receptivity on the day of anthesis. In citrus, both staminate and hermaphrodite flowers are produced on the same plant. Floral biology constitutes flowering period, flower bud differentiation, flower bud development, inflorescence, sex ratio, anthesis, dehiscence, pollen fertility and stigma receptivity. Then now here we will see the inheritance characters of citrus. Like first, leaf character governed by two principal genes. Anthocyanin pigment governed by dominant gene controlling this reddish color. Anthocyanin more than one recessive gene are involved in case of fruit. Dominant gene in case of leaf but recessive gene in case of fruit. Tristeza virus, single allele, texture governed by many genes. So that's why polygenic control. Polyembryony more than one single gene. Seedless nature governed by recessive gene and nucellar embryony that is governed by one or more dominant gene. Next, flower bud development. In various forms of citrus, the complete development of bud lasts from 18 to 47 days. In northern India, sweet orange flower bud takes 18 to 22 days, while in lemon 20 to 32 days. In sweet lime, it takes 22 to 25 days. So just this you need to keep in mind. The development of flower bud in citrus is greatly influenced by the prevailing temperature. As temperature increases, the time taken by bud to development is reduced. In citrus, 7 stages of bud development are observed. So stage 1, buds have just appeared. They are tiny, round, completely covered by calyx lobe, hence green in color. Say stage 2, the buds are still round, purple in color. Corolla just started bulging and calyx still prominent and anther will be green. So in stage 2, microsporogenesis will occur. So stage 3, bud conical in shape due to elongation of corolla. Deep purple color, length of corolla as well as calyx cup now become equal but anther still green. Stage 4, bud will enlarge from conical shape to elliptical oblong. So length of corolla almost double to length of calyx. Anther become yellowish green. So stage 5, further elongation of corolla, elliptical and oval. Length of corolla lobe becomes approximately 3 times of calyx. Anther light yellow, stigma not fully developed, usually greenish in color and furrowed. In stage 6, buds attain full size and shape. According to varieties and length of corolla lobes become 3 times equal to its calyx and anther light yellow. Stigma yellow. Buds soft, plumpy and ready to open on the next day. Stage 7 means the flower will open fully. So see here the buds and flower picture. Right. Then next, inflorescence. So this inflorescence in different citrus fruit is of cymose type or corymbose racemes. Inflorescence develop from dormant axillary vegetative buds. Inflorescence formation was under genetic control and these inflorescence patterns were influenced by environmental condition after bud break. The flowers in citrus are borne on 2 types of shoots. So first, then the next type aspect, sex ratio. Perfect flowers is higher in the beginning. Low temperature and relatively high humidity seems to be more conducive. Leafy inflorescence produce large number of hermaphrodite flowers. Genus Poncirus, citrus lemon, citrus argentifolia, citrus medica and related types, the pistil is underdeveloped or absent in large percentage of flowers. In Citrus sinensis, Citrus grandis, Citrus paradisi and Citrus reticulata, abortion of pistil is much less frequent. Anthesis. More than 80% flower open before one afternoon. Factors affecting anthesis, that is temperature, light, position of flower bud, type of inflorescence and aspect of that particular tree. Terminal buds open earlier than the lateral ones. Flowers on leafless inflorescence open earlier than those on leafy inflorescence. Flowers on shade side of the tree open later than those exposed to sunshine. Dehiscence. Dehiscence of anthers in citrus takes place at different times. Some cultivars are protandrous lemon, while some are protogynous, that is sweet lime and pomelo. High temperature accelerates the dehiscence of anthers. Normal anthers are bright yellow at maturity. So anther containing no pollen are pale green or white and usually do not dehisce. Then stigma receptiveness. So in most of the citrus species, the androecium and gynoecium matures at the same time, but some species are protandrous. The stigma is receptive from 1 to few days before anthesis and for several days afterwards. The prevailing weather condition influences duration of stigma receptiveness. Next, storage, longevity and fertility of pollen. So trifoliate orange pollen remains viable for 110 days when stored in desiccator over calcium chloride. Kaxi lime and grapefruit pollen can be kept for 37 to 38 months at a temperature of 0 degree centigrade and 25% relative humidity. Pollen storage by home freezer can be the most convenient method for storing the pollen for short duration. Powdered milk, non-fat, egg albumin and lycopodium are some of the pollen diluents. Percentage of functional pollen varies greatly among species and cultivar in case of citrus. So Washington navel, no viable pollen. Satsuma mandarin, very little. Most of the cultivars of lemon and orange, more than 25% viable pollen. So most mandarin and pomelo cultivars have high percentage of functional pollen. Next, pollen germination. The germination range from 16% in Valencia to more than 90% in several other varieties. In Morton citrons, the germination is obtained with 10% sugar solution with 58 gram agar-agar in water. So 15% sugar solution to be optimum for germination for different lemon and citron pollen at 20 degrees centigrade. 20% sugar solution gives maximum germination at 28 degree in sweet lemon. So this is the chemical composition of culture media for in vitro germination of pollen. Like these are the chemicals like agar, if solid media 1%, sucrose, solid liquid media 15%, H3BO3 100 ppm in solid and liquid. So CaNO3 by 2 plus 4H2O. When solid media 300 ppm and in liquid 1000 ppm. Magnesium sulfide with water 200 ppm in solid media and 300 in liquid media. So KNO3 100 ppm in solid and liquid. Desirable attributes of scion cultivars. We know that in citrus breeding we will use scion and rootstocks. So now first we will see desirable attributes of scion and after that we will see desirable attributes of rootstocks. Desirable for scion, desirable fruit size, shape, exterior appearance, tight skin with pleasant flavor. Vigor and longevity of the tree with sufficient amount and regularity of the crop. Reduced tree size without the reduction of yield per unit canopy volume to facilitate easy handling of the plants. Seedless or less seeded fruits. Season of ripening early or late. Storage life and adaptability to specific environments. Feasibility to mechanical harvesting. Compatibility with important rootstocks. Next, desirable attributes of rootstock. Reduction of tree size without affecting the yield or scion health. Compatibility with the important scion cultivars. Resistant to citrus and burrowing nematodes. resistant to root rot disease caused by Phytophthora, which seems to take a heavy toll of seedlings in the nursery and grown-up trees in the field. Tolerance to excess salts, drought, cold and waterlogging. Rapid growth and lack of branching so that it can be handled easily in the nursery. Now we'll see what are the different breeding methods. So first, introduction. So introduction of germplasm either from other countries or from one agroclimatic region to the other. One agroclimatic region to other within the country. That has been one of the most potent improvement method. The mandarin variety Santra is known to have been grown in India for many centuries. It was introduced into the central provinces, that is now Maharashtra, by Raghuji Bhosle II from Aurangabad in 18th century. Right. Tangerines. So this spelling. Yeah. So in 18th century it was introduced. Tangerines, St. Michael blood orange and large white orange were imported and cultivated in Gujranwala in Punjab during 1880. The present century has seen the introduction of number of sweet orange varieties including Washington Navel, Valencia, Jaffa, Blood Red Malta and Tangerines. The first two were introduced from America and the others from respective countries of their origin. Grapefruits were introduced from California and Florida, lemons from China and Malta, from USA and Italy. Mossambi seems to have been introduced in Nagpur during the beginning of 20th century. The introduction of Quino mandarin, that is king cross with willow leaf, in 1947 showed great promise in North India. It was introduced in South India in 1958 and Punjab in 1959 and has performed extremely well in Punjab. Next method, that is clonal selection. Exploitation of natural variability existing in a variety has resulted in the isolation of some promising clones in citrus. PKM1 lime is a clonal selection from seedling progenies of Kadayam type of Tirunelveli district of Tamil Nadu. Yuvraj blood red is a seedless and early maturing clonal selection from blood red orange. Pramalini and Vikram, the two Kaxi lime varieties, were developed through clonal selection at MAU Parbhani. Chakradhar is a thornless and seedless selection from Kaxi lime. So see, these are the some selections in citrus. So orange, Yuvraj blood red from blood red, mandarin, Madhukar seedless from Nagpur Santra. In case of these are both from MAU Parbhani. In case of acid lime, Pramalini, Vikram and Jaydevi, that is PKM1. So all three selected from acid lime by MAU Parbhani. Seedless selection from acid lime selected at Himachal Pradesh. Chakradhar lime from Kaxi lime, Periyakulam, Tamil Nadu. Tenali from acid lime, Tirupati. Saisarbati from acid lime from Rahuri. Then lemon PAU selection at PAU. Davwali Baramasi PAU. Pant lemon Pantanagar, Kaxi Kalan at Saranpur. So hybridization. Next method is hybridization. Hybridization is confronted with the real problems in citrus improvement, both on scion as well as rootstock, because the long juvenile phase delays the assessment of hybrids. Most of the cultivated varieties are highly polyembryonic. Hence, the crosses made using this as a females results in very few weak hybrids, which are difficult to identify from nucellar seedlings. Electrophoretic techniques separating the isozymes of parents and hybrids may be of great value in scion breeding program, as no morphological markers are available at present. The heterozygous nature of the crop further leads to wide segregation. The problems are little less complicated of rootstock breeding, where Commonly used disease-resistant male parent, Oncerus trifoliata, has trifoliate leaves which is dominant over the monofoliate character. So in other citrus varieties and all hybrids, by which distinction of the unifoliate nucellar seedlings could be easily made. Right. Next, hybridization techniques. So the mature flower buds of the female parent are emasculated early in the morning on the day of opening and are packed. The flowers to be used as a male parent are packed the previous evening. The next morning, as the temperature rises, the anthers dehisces, releasing pollen. These flowers are then plucked and used to pollinate the receptive stigmas of the emasculated flowers. The pollinated flowers are again back, open after about a week and allowed to develop into mature fruits. In some cases, especially when trifoliate orange is used as a male parent, difficulties arise because of its flowering finishes earlier than other citrus varieties. Therefore, pollen must be stored under low humidity and temperature conditions. Seeds from mature fruits are extracted and sown immediately in sterilized sand soil mixture. When seedlings reach about 15 cm height, hybrid seedlings are identified. Seedlings showing morphological characters of male parent are selected, while remaining things you need to discard. Electrophoresis technique can also be used to identify the zygotic seedlings. Hybrid seedling identification and rootstock evaluation. Identification of hybrid seedlings having trifo as a male parent is easily done by observing the trifoliate leaf character. Hybrid seedlings are grown to maturity in the field and their progenies are evaluated for resistance to diseases, insect pests, nematodes, and suitability as a scion or rootstock. Then evaluation for rootstock purpose. Rootstock hybrids should possess the desirable traits such as high percentage of nucellar embryony, resistant to diseases and nematodes. So selected hybrids are tested with different scion varieties as compared with the standard commercial rootstocks. Important observations include plant growth, fruit characters, yield and yield contributing traits. Evaluation of scion hybrids. Like in the first round of evaluation, zygotic seedlings are raised on suitable rootstocks and observations on various vegetative and fruit characters you need to record. Resistant to different diseases is also confirmed. Selected hybrids are further tested on different rootstock across locations and compared with commercial varieties. So intergeneric and intrageneric hybrids. So first intergeneric. Although intergeneric hybrids are generally rare in fruit crops, considerable success has been achieved in citrus. Polyploidy breeding. Most of the species and varieties of citrus are diploids, but occurrence of polyploidy has been reported in many cultivars. The Hong Kong wild cumotes Fortunella hentzi may have been the first reported tetraploid. Polyploidy breeding seems to offer prospects to obtain large size fruit with dwarf plant types. The production of triploids by crossing the tetraploid with diploid may be useful in obtaining the seedless varieties. The seedless line, that is Citrus latifolia, is a triploid. Triploids have favorable characteristics and yield. Well, they are sterile. The development of triploid through breeding is very limited. Production of triploid is normally achieved by crossing the tetraploid with diploid, which is often not feasible. The reciprocal cross produces many tetraploid individuals. Polyploidy manipulation by crossing of tetraploids and with diploids yielded some valuable triploid varieties like Orlando and Mellow Gold. A large diversity of autotetraploid parents with desirable characters expressed in the progeny will be of high value to any citrus cultivar breeding program. Spontaneous autotetraploids occur among many polyembryonic citrus varieties. Tetraploid trees of monoembryonic varieties can be obtained by colchicine treatment. Then next, biotechnology in improvement of citrus. Transformation of fruit species by biotechnological tools is a potential approach to develop disease-resistant cultivars. Woody plants are also known to be difficult to work in vitro than herbaceous plant, but citrus is exceptional. Though nucellar embryony in citrus is of great value for producing vigorous, uniform and virus-free plants, it appears to be an obstacle in hybridization. In polyembryonic cultivar, the vigorous growth of nucellar embryos inhibit the growth of the zygotic embryo and causes its degeneration. Right? Prior to seed maturation. Such abortive embryos can be rescued by tissue culture. Tissue culture has effectively been used in obtaining hybrid Poncirus plantlets from polyembryonic citrus cultivar. Poncirus trifoliata not only carries a genetic marker but also possesses resistance to Tristeza, Phytophthora, nematode and cold stress. Intergenetic hybridization with the aid of cell tissue culture offers the possibility of incorporation of multiple desirable characters found in genera for improvement of citrus rootstock and scion cultivars. Cell and tissue culture and especially protoplast manipulation have effectively been explored in citrus improvement by regeneration of citrus trees from protoplasm, somatic hybridization. That is also called as hybridization and organelle transfer. In an attempt to develop a protoplast derived plants, in the last 1 decade, Israel and Florida have shown the protoplast system in the dozen genera. And interestingly, citrus is the only woody plant among them. Efficient protocols have been developed to obtain protoplast with cell division capability from all the major citrus cultivar and some of their wild relatives. So now here we will see some important species and cultivars of mandarin. So first, Citrus reticulata, then calamondin, that is Citrus madurensis, with fruit color orange to deep orange, smooth and glossy surface, pitted shape, oblate deep orange and size small, with flattened base having 7 to 10 segments. Next, clementine, that is Algerian tangerine. It is a tangerine and is probably an accidental hybrid of mandarin and sour orange, which is considered to be originated in Algeria. Fruit color deep orange, shape is globose to elliptical, size medium with depressed apex. Segments 8 to 12. It is an early variety. Then Cleopatra, that is Citrus reshni, originated in China. Plant is thornless with dense top. Fruits are produced singly or in cluster. Fruit color dark orange red. Shape oblate. And segments, small segments, size is small and segments 12 to 15. Next, Kurg orange. It is an important variety of South India, particularly in Kurg and Wayanad tracts. Fruits are medium to large, bright orange color, oblate to globose in shape, finely papillate and wrinkled, glossy and 9 to 11 segment. Next, Dancy tangerine. In USA, the Dancy is the best known and highly prized of all the mandarin oranges. Tree large, nearly thornless and has an upright growth. Fruit color deep orange red to scarlet. Rind thin, loose, easily separable. Segments 10 to 14. It is late variety. Next, Desi mandarin, Pathankot. This variety is mainly grown in Punjab hills. The tree is large with semi-upright growth habit and compact foliage and are spineless. Fruit are ovoid to soft globose. Color uniformly cadmium, surface pitted, semi-glossy and finely wrinkled. Rind medium. Adherence slight. Segment 7 to 10. King mandarin. Swingle believed the king mandarin has a tang, a hybrid between mandarin and sweet orange. King mandarin was first introduced from Cochin, China to California in 1882. King mandarin is cultivated in Assam. This is the prolific bearer, frost resistant and produces high quality fruit. Next, Khasi mandarin, that is Citrus nobilis. It is commercial variety of Assam. This is believed to be a hybrid between mandarin and sweet orange. So fruit is depressed. Globose to oblate, orange yellow to bright orange. Smooth surface, prolific bearer, frost resistance. Next, willow leaf mandarin, that is Citrus deliciosa. Tree is willow in growth. Fruit color orange. 10 to 12 segments. It is an early variety. Next, Kinno mandarin, king cross with willow leaf mandarin. It is a first generation hybrid between king and willow leaf mandarin and developed by H.B. Frost at the California Citrus Experiment Station in the year 1915. It was introduced into Punjab from USA. It is late. Then fruit color resembles of king, that is deep yellowish orange. 9 to 10 segments you can easily separate. 12 to 24 seeds. Next, well known Nagpur Santra. So this variety occupies prime position in Indian market and is one of the finest mandarin grown in the world. It is also known as Ponkan. Tree is large, vigorous and spineless with compact foliage. Fruit is medium, cadmium color, smooth surface and glossy. Next, Satsuma orange. It is Japanese variety introduced into Florida. It is frost resistant and useful as a breeding material. It is also resistant to canker, gummosis and scaly bark. Plant is thornless, having spreading habit. Right. And most important, it is rich in flavor and seedless. Next, temple mandarin. It is a hybrid between tangerine and sweet orange. Temple mandarin is most beautiful and highly flavored fruit of the citrus group. So tree is medium, thorny, deep orange to reddish fruit color. Orange pulp. So it is late in maturity. Then in case of lemon, that is citrus lemon, Eureka. It is the seedling selection of Cilician lemons. Tree is medium, spreading, thornless. Fruit color lemon yellow. 8 to 10 segments. Size is medium. Eureka is heavy yielder. Begins bearing at early stage and tendency of bearing in the terminal end of the shoot. Next, Lisbon. Its appearance and yield is superior to Eureka, resistant to frost, heat and high wind velocity. Tree is large, vigorous with spreading shoots. 6 to 10 segments and 0 to 8 seeds. Punt lemon. Fruit size medium, juicy, heavy fruiting. Tolerant to pest disease. Then next, Villifranca belongs to Eureka group and was introduced into Florida from Europe. Tree is vigorous, thorny, spreading fruit oval to oblong, medium size, color bright lemon yellow. Segment 8 to 12, juice colorless and 25 to 30 seeds. Then next, Mare lemon. Tree semi-dwarf. Thornless, spreading, cold resistant, fruit color light orange. Surface is smooth, finely pitted. 8 to 10 segments and 8 to 12 seeds. Lucknow seedless. It is hardy, medium, vigorous, spreading, drooping. 10 to 12 segments. Fruit color is yellow and maturity during November and December. Next, Nepali oblong. Plant medium size, hardy, spreading, fruit shape oblong, color is lemon yellow, 8 to 10 segments and 8 to 12 seeds. Hill lemon, galgal, that is citrus, pseudo lemon. It is indigenous to North India, grown in sub-Himalayan region. Plant is tall, vigorous, upright and spreading with an irregular and loose crown. Fruit ovate oblong, yellow. Base round, 7 to 11 segments, 25 to 30 seeds. Ripening during October to December. Lemon hybrids. So here you can see some citrus products hybrids. Then lemon hybrids first, lemonades, lemon crossed with sweet orange. Then lemonim, lemon with lime. Lemandrin, that is lemon with mandarin. Iremo lemon, that is Australian desert crossed with lemon. Sweet orange, Citrus sinensis. moderate to high TSS, low to moderate acid. So there are four classes: common round orange, navels, pigmented orange, acid-less or sugar orange. Based on maturity, three classes: early, mid and late. Early 6 to 9 months, mid 9 to 12 months and late more than 12 months. Then last, genetic variability. A wide range of variability exists in citrus with respect to fruit morphology and quality. Citrus fruits ranges in size from very small to very large. Among the smallest are kumquats, that is Fortunella species, and the lime, Citrus aurantifolia. And the other extreme like Pumelo, Citrus grandis and Citron, Citrus medica. Fruit range color ranges from yellow-green of the limes to red-orange of the mandarins. Fruit shape range from oblate to pyriform. So at maturity, fruits of some cultivars are very high in acid, while others have almost none. So several man-made and natural hybrids are now available for breeding program. So see here the variability in citrus fruit, like small, extreme big, pale yellow, then deep red. So these are the genetic variability present in citrus. So this is about advances in citrus breeding. Right from where the classification, breeding objectives, breeding methods, some varieties and genetic variability. Hope you will get some valuable information from this session. Okay. So till next session, thank you very much.